What do the structure of DNA, the windshield wiper, Wi-Fi, Kevlar, Monopoly, and modern antiviral medicine have in common? They were all shaped by women. Women who discovered something no one had seen before. Women who invented technologies used by billions of people. Women who created art, stories, and industries. And yet in many of these stories, something strange happened. The discovery survived. The invention spread. The industry grew. But the woman behind the idea sometimes disappeared from the credit line. Today, we are putting those names back where they belong, because innovation history looks very different when you read it carefully.
You are listening to Intangiblia, the podcast of intangible law, plain talk about intellectual property. Please welcome your host, Leticia Caminero.
Welcome back to Intangiblia. I'm your host, Leticia Caminero. Today, we are celebrating International Women's Day with a special episode about women who changed the world through ideas, science, technology, medicine, art, entrepreneurship. When we look through the lens of intellectual property and innovation history, we find extraordinary stories of women whose work transformed entire industries. Some of them received recognition immediately, others waited decades. Some had to fight to reclaim authorship. But every one of them changed the trajectory of knowledge. Today, we are walking through 16 stories that show how women helped build the modern world. Before we begin, a quick note. The voice you are hearing is an AI-cloned voice of the real Leticia Caminero.
And I am Artemisa, a fully AI co-host designed to help unpack intellectual property stories.
This episode is for educational storytelling purposes.
It is not legal advice.
Let's begin with one of the most famous scientific discoveries in human history, the structure of DNA. In the early 1950s, scientists around the world were racing to understand the molecular structure of the substance that carries genetic information. Whoever solved it would unlock one of the deepest mysteries of life itself.
At King's College London, a brilliant chemist named Rosalind Franklin was using a technique called X-ray crystallography. This method allows scientists to analyze the structure of molecules by observing how X-rays scatter when they pass through crystallized samples.
Franklin was extraordinarily precise. She insisted on rigorous experimental methods and careful interpretation of data. In 1952, she produced a photograph that would become legendary. It was known as Photo 51.
And if you look at Photo 51 today, even non-scientists can see something remarkable. A pattern that clearly reveals a spiral structure, the double helix.
Franklin's image contained crucial evidence showing that DNA had a helical structure with specific repeating dimensions. Her work also allowed scientists to calculate the spacing between the molecular components.
But something controversial happened. Without Franklin's permission, the photograph and some of her unpublished data were shown to two scientists working on the same problem at Cambridge, James Watson and Francis Crick. Using that information, Watson and Crick constructed their famous model of the DNA double helix in 1953.
And that model became one of the most iconic scientific breakthroughs of the 20th century, the foundation of modern genetics.
In 1962, the Nobel Prize in Physiology or Medicine was awarded to Watson, Crick, and Maurice Wilkins. Rosalind Franklin was not included.
Partly because Nobel Prizes are not awarded posthumously. Franklin had died in 1958 at only 37 years old, but historians later argued that her contribution was essential to the determination of DNA's structure.
Today, Franklin is widely recognized as a central figure in the discovery. Her work is often discussed as a classic example of the Matilda effect, a term used to describe how women's scientific contributions have historically been overlooked or attributed to male colleagues.
The term was introduced in 1993 by historian of science Margaret Rossiter. She was studying the history of women in science and kept finding the same pattern again and again. Important discoveries, major contributions, but incomplete recognition.
Rossiter named the phenomenon after Matilda Joslyn Gage, a 19th century writer and activist who had already noticed this problem more than 100 years earlier. In 1870, Gage wrote about how women inventors and scientists were frequently ignored or written out of the story.
So Rossiter connected the dots and gave the pattern a name, the Matilda effect.
And when you start looking back at the history of science, you realize that many discoveries we learn about today were actually the work of teams. And sometimes the women in those teams were the ones who made the critical breakthrough.
And several of the stories in this episode show why understanding that pattern still matters today.
Our second story takes us into the world of nuclear physics and one of the most consequential discoveries of the 20th century, the discovery of nuclear fission. At the center of this story is Lise Meitner, an Austrian-Swedish physicist whose work helped explain how the nucleus of an atom could split into smaller parts and release enormous amounts of energy.
In the early 20th century, Meitner was already an accomplished scientist. She had spent decades studying radioactivity and nuclear reactions, often working alongside the German chemist Otto Hahn in Berlin. For more than 30 years, they collaborated on experiments that explored how heavy elements behave when bombarded with neutrons.
And remember, this was an era when women in physics were extremely rare.
Many universities did not even allow women to hold full academic positions.
Despite these barriers, Meitner became one of the leading nuclear physicists of her generation.
Then history intervened. In 1938, because she was Jewish, Meitner was forced to flee Nazi Germany. She escaped to Sweden, leaving behind her laboratory, her equipment, and the experimental work she had spent decades building.
But the collaboration with Hahn did not end entirely. Later that year, Hahn conducted experiments that produced puzzling results. When uranium atoms were bombarded with neutrons, the resulting elements were much lighter than expected. Hahn wrote to Meitner asking for help interpreting the results.
Which is where the breakthrough happened. While walking in the snow during a holiday visit with her nephew, physicist Otto Frisch, Meitner worked through the calculations. She realized that the uranium nucleus was not simply rearranging itself, it was splitting.
Meitner and Frisch developed the theoretical explanation showing that the uranium nucleus had undergone a process they called fission, releasing an enormous amount of energy predicted by Einstein's equation E equals mc squared. Their paper explaining nuclear fission was published in 1939. At the same time, Hahn published his experimental results.
And then came the Nobel Prize. In 1944, the Nobel Prize in Chemistry was awarded solely to Otto Hahn for the discovery of nuclear fission. Meitner was not included.
Many historians and scientists later argued that this decision overlooked Meitner's essential role in explaining the phenomenon. Without her theoretical interpretation, the experimental observations would have remained unexplained.
In other words, Hahn observed the results. Meitner explained the physics that made them meaningful.
Which is not a small contribution. It is the difference between seeing something strange and understanding a fundamental new process in nature.
Today, Meitner is widely recognized as one of the key figures behind the discovery of nuclear fission. She later became known as the mother of nuclear physics, although she herself opposed the use of nuclear weapons. Interestingly, the element meitnerium, discovered in 1982, was named in her honor.
So even if recognition came late, science eventually corrected the record.
Discovery is rarely the work of a single individual. It is the product of collaboration, ideas, and insight. The challenge is making sure that everyone who contributed is properly remembered.
Our third story takes us into the universe. Literally. In 1967, a young astrophysics graduate student named Jocelyn Bell, later known as Jocelyn Bell Burnell, was working at the University of Cambridge on a large radio telescope designed to study distant cosmic signals. The telescope was not small.
It stretched across several acres of land and consisted of thousands of wires and wooden poles. Bell herself helped assemble the equipment and was responsible for analyzing the massive amounts of data the telescope produced. And when I say massive, I mean truly massive.
Every day, the telescope produced meters of chart recordings. which meant her job involved a lot of staring at long sheets of paper, looking for patterns. Not exactly glamorous science, but sometimes the biggest discoveries start exactly that way.
One day, Bell noticed something unusual in the data, a tiny signal that appeared as a very regular pulse. At first, it looked like interference, maybe a problem with the equipment, but the signal was incredibly precise. It repeated every 1.337 seconds, over and over again. Bell began investigating further and eventually identified multiple sources of these repeating signals. They were coming from deep space.
And because the pulses were so perfectly regular, the research team jokingly nicknamed the source LGM, short for Little Green Man.
Of course, the explanation turned out to be even more fascinating. The signals were coming from a new type of astronomical object that had never been observed before.
Pulsars. A pulsar is a rapidly rotating neutron star, the collapsed core of a massive star that exploded in a supernova. These objects spin incredibly fast and emit beams of electromagnetic radiation from their magnetic poles. When those beams sweep past Earth, they appear as regular pulses of radio waves.
Bell's careful attention to the data revealed the first known pulsar. It was one of the most important astronomical discoveries of the 20th century.
Which brings us to the moment where recognition enters the story. In 1974, the Nobel Prize in Physics was awarded for the discovery of pulsars. The prize went to Anthony Hewish, Bell's supervisor, and radio astronomer Martin Ryle. Jocelyn Bell Burnell was not included.
The decision sparked debate in the scientific community. Many scientists believed Bell's role in identifying the signal was crucial to the discovery. After all, she was the one who first noticed the anomaly and pursued it instead of dismissing it as noise.
Which, in science, is often the moment where discovery actually happens. Recognizing that something strange in the data might mean something important.
Bell Burnell herself responded to the situation with remarkable grace. She often explained that Nobel Prizes typically go to senior researchers rather than graduate students. But historians of science continue to point out that her role was central to the discovery.
Over time, Bell Burnell received many other major honors. In 2018, she was awarded the Breakthrough Prize in Fundamental Physics, worth $3 million.
In an extraordinary gesture, she donated the entire prize to fund scholarships for women, minority students, and refugees pursuing physics.
Which might be one of the most powerful endings to a scientific story. The discovery of pulsars not only changed astrophysics, it helped open the door for future generations of scientists.
And from an intellectual property perspective, the lesson is clear. Innovation often begins with curiosity, the willingness to notice something unusual, investigate it, and ask the question others might overlook.
In this case, curiosity revealed a new kind of star and expanded our understanding of the universe.
Our next story is about a discovery that overturned one of the most fundamental assumptions in physics. For decades, physicists believed that the laws of nature were perfectly symmetrical. In particular, they believed in a principle called parity conservation.
Parity means that the laws of physics should behave the same way if you look at a system in a mirror. Imagine watching a physical process, then watching its mirror image. According to parity conservation, both should obey the same rules. For many years, scientists assumed this symmetry was universal.
But in the 1950s, two theoretical physicists, Tsung-Dao Lee and Chen-Ning Yang, began to suspect something unusual. They proposed that parity might not hold true in certain types of particle interactions, specifically in weak nuclear interactions. It was a radical idea, but there was a problem. They needed experimental proof.
Which is where Chien-Shiung Wu enters the story. And if you ask many physicists, this is where the real breakthrough happened.
Wu was already one of the most respected experimental physicists in the world. She had built a reputation for designing extremely precise experiments in nuclear physics. Lee and Yang approached her with their theoretical idea.
Wu immediately understood the challenge. Testing parity violation required an experiment of extraordinary precision. It involved cooling radioactive cobalt atoms to extremely low temperatures and observing how electrons were emitted during radioactive decay. If parity was conserved, the emissions should appear symmetrical. If parity was violated, the pattern would break that symmetry.
Wu designed and carried out the experiment at the National Bureau of Standards in the United States. The results were stunning. The electron emissions clearly showed that parity was not conserved in weak interactions. In other words, the universe itself was not perfectly symmetrical.
Which meant a core assumption of physics had just been overturned. Not a small adjustment. A complete shift in how scientists understood fundamental forces.
The experiment became known as the Wu experiment, and it fundamentally changed particle physics. In 1957, the Nobel Prize in Physics was awarded to Li and Yang for their theoretical work predicting parity violation. Chien-Shiung Wu was not included in the prize,
even though she designed and conducted the experiment that actually proved the theory.
Many physicists later described this as one of the most significant Nobel omissions in modern science.
Wu continued to have an extraordinary career. She became one of the first women to serve as president of the American Physical Society and received numerous scientific honors.
She was sometimes referred to as the First Lady of Physics.
Which sounds elegant, but it also hints at something deeper. Wu was not simply a symbolic figure. She was one of the most technically skilled experimental physicists of her generation.
And her story highlights an important lesson about innovation. Breakthroughs rarely come from theory alone. They depend on people who can translate ideas into experiments, evidence, and proof.
In many ways, that experimental validation is what turns a hypothesis into a discovery. In the case of parity violation, that turning point was the work of Chien-Shiung Wu, a scientist whose experiment reshaped the laws of physics.
Our fifth story takes us into the microscopic world of bacteria and into one of the most important scientific revolutions of the 20th century, the rise of modern genetics. At the center of this story is Esther Lederberg, a brilliant microbiologist whose discoveries helped transform how scientists study genes and microorganisms.
In the 1950s, genetics was entering a new era. Scientists were trying to understand how genes function, how they move, and how they influence biological processes. Esther Lederberg made several discoveries that became foundational to the field.
One of her most important contributions was the discovery of a virus that infects bacteria, known as the lambda bacteriophage. This virus became one of the most important tools in molecular genetics. Researchers use it to understand how genes replicate, mutate, and regulate biological processes.
In other words, this was not a small laboratory curiosity. It became a core model organism for studying genetic mechanisms.
But that was only one of her contributions. Esther Lederberg also developed a technique called replica plating, a method that allows scientists to transfer colonies of bacteria from one petri dish to another in exactly the same spatial pattern. This method made it possible to study mutations and antibiotic resistance in a controlled and repeatable way. Replica plating became a fundamental tool in microbiology labs around the world.
Which sounds simple now, but at the time it was a breakthrough method that allowed scientists to observe genetic changes with unprecedented precision.
These discoveries were deeply influential in the emerging field of molecular biology, but recognition did not arrive in equal measure. Esther Lederberg's husband, Joshua Lederberg, was also a geneticist and collaborator in related research. In 1958, Joshua Lederberg received the Nobel Prize in Physiology or Medicine for work on bacterial genetics. Esther Lederberg was not included,
which has led many historians of science to revisit the story, because several of the tools and discoveries that shaped bacterial genetics were directly linked to her work.
Beyond the Nobel question, Esther Lederberg also faced significant institutional barriers. Despite her scientific accomplishments, she struggled to obtain the same academic recognition and positions that male colleagues received. Her contributions were often treated as supporting work rather than as independent scientific leadership.
which is remarkable when you consider how central her discoveries became to modern biology. Lambda phage, replica plating, microbial genetics. These are not minor footnotes. They are core building blocks of the discipline.
Today, historians and scientists increasingly recognize Esther Letterberg as a pioneer in microbial genetics. Her work helped establish the experimental tools that made modern genetic research possible. From an intellectual perspective, her story reminds us that innovation is often collaborative. Scientific breakthroughs rarely emerge from a single mind. They grow from networks of ideas, experiments, and discoveries.
Ensuring that every contributor receives proper credit is essential, not only for fairness, but also for preserving the true history of science.
Our sixth story takes us back to the early 20th century and to a question that scientists had debated for centuries. What determines whether an organism develops as male or female? For a long time, the explanations ranged from environmental conditions to maternal influence. Some scientists believed temperature or nutrition played a role. Others believed the answer lay somewhere in embryonic development. But no one had definitive proof.
Enter Nettie Stevens, an American geneticist working at the Carnegie Institution and Bryn Mawr College. Stevens was studying insects, particularly a species of beetle called Tenebrio molitor, also known as the mealworm beetle.
Using careful microscopic analysis, she began examining the chromosomes present in the reproductive cells of these insects,
which at the time was cutting-edge research. Remember, genetics was still a young science. Scientists had only recently begun to understand that chromosomes carried hereditary information.
While analyzing the beetle cells, Stevens noticed something remarkable. Female beetles had 2 large chromosomes in a particular pair. Male beetles had 1 large chromosome and 1 much smaller one.
That difference was consistent across specimens. Stevens concluded that these chromosomes determined biological sex.
The larger chromosome became known as the X chromosome, and the smaller one became the Y chromosome.
In other words, she demonstrated that sex is determined by chromosomal combinations. XX produces female offspring. XY produces male offspring.
This discovery provided the first clear scientific evidence linking sex determination to specific chromosomes. It was a major breakthrough for genetics and biology.
Stevens published her findings in 1905. Around the same time, another scientist, Edmund Beecher Wilson, published related work on chromosomes and sex determination.
Over time, Wilson often received more recognition in textbooks and scientific histories.
Which is surprising because Stevens' experimental work was extremely precise and arrived at the same conclusion independently. Some historians argue that her contribution was actually more definitive.
Unfortunately, Stevens died in 1912 at the age of 50, relatively early in her career. Because of that, and because of the scientific culture of the time, her work did not receive the full recognition it deserved for many years. Today, geneticists widely acknowledge Nettie Stevens as one of the scientists who discovered the chromosomal basis of sex determination. Her research helped establish the foundation of modern genetics,
which means that every time biology students learn about X and Y chromosomes, they are learning a discovery that traces back to her microscope.
Our seventh story is about a discovery that initially sounded so strange that many scientists simply refused to believe it. The discovery of jumping genes.
The scientist behind it was Barbara McClintock, an American geneticist working in the mid-20th century. McClintock specialized in studying maize or corn, which at the time was a powerful model organism for genetic research. By analyzing the chromosomes of maize plants under a microscope, she began observing patterns that did not fit the traditional understanding of genetics.
At that time, scientists believed genes were fixed units, sitting in stable positions along chromosomes. Genes were supposed to stay in place. They did not move. But McClintock's experiment suggested something completely different.
She found evidence that certain genetic elements were actually moving within the genome, which sounded almost impossible at the time. Genes were supposed to behave like coordinates on a map, not like travelers.
While studying color patterns in maize kernels, McClintock noticed that some genes appeared to switch on and off, depending on their position within the chromosome. After years of meticulous research, she proposed that some genetic elements could transpose, meaning they could move from one location in the genome to another. She called these elements controlling elements. Today we call them transposable elements, or more informally, jumping genes.
Which, if true, would mean the genome was not a static blueprint. It was dynamic, flexible, almost like a system that could rewrite parts of itself.
McClintock published her findings in the early 1950s, but the scientific community reacted with skepticism. Many researchers believed the idea was simply too radical. At the time, there was no clear molecular explanation for how genes could move. As a result, McClintock gradually stopped publishing on the topic and continued her research quietly.
Which is the scientific version of saying, the world is not ready for this yet.
Decades later, advances in molecular biology began to confirm her observations. Scientists discovered that transposable elements were real and that they play important roles in gene regulation, genome evolution, and genetic diversity. Today, we know that a large portion of many genomes consists of transposable elements. In humans, for example, they make up a significant percentage of our DNA. In 1983, Barbara McClintock received the Nobel Prize in Physiology or Medicine for her discovery of mobile genetic elements,
which made her the first woman to receive an unshared Nobel Prize in that category.
Her recognition arrived more than 30 years after her original discovery. But when it came, the scientific community acknowledged the profound impact of her work. McClintock's research changed how scientists understand genomes. Instead of being static structures, genomes are now seen as dynamic systems capable of rearranging themselves over time.
Which means that the concept of genetic mobility is now fundamental to fields like evolutionary biology, biotechnology, and medicine.
And from an intellectual innovation perspective, McClintock's story highlights an important lesson. Sometimes the most groundbreaking discoveries are not rejected because they are wrong. They are rejected because they are too far ahead of existing scientific frameworks. Barbara McClintock had the patience and confidence to trust her data, and eventually the world caught up with her discovery.
Intangiblia, the podcast of intangible law, plain talk about intellectual property.
Our eighth story is one of the most powerful and heartbreaking in the history of medical innovation. It is the story of Alice Augusta Ball, a chemist whose work transformed the treatment of leprosy, also known as Hansen's disease. At the beginning of the 20th century, leprosy was one of the most feared diseases in the world. Patients were often isolated in colonies, separated from their families and communities.
There was no reliable cure. Doctors had experimented with chalmugra oil, a natural oil extracted from the seeds of certain trees. The oil had shown some promise in treating the disease, but it had a major problem. It was extremely thick and difficult to administer.
Patients were often forced to drink it, which caused severe nausea, or it was applied topically with inconsistent results. Injecting the oil directly into the bloodstream seemed like a possible solution, but its chemical properties made that almost impossible.
In other words, the treatment existed, but the chemistry made it unusable. And that is where Alice Ball enters the story.
Alice Ball was a young chemist at the University of Hawaii, and she was only in her early 20s when she began working on the problem. Her challenge was to modify chalmugra oil so that it could be injected safely into the body. Ball developed a chemical process that isolated the active compounds in the oil and converted them into water-soluble ethyl esters.
This transformation allowed the medication to be injected into patients effectively. The treatment became known as the Ball method. For the first time, patients suffering from leprosy could receive a treatment that significantly improved their condition and allowed many to leave isolation colonies.
Which is extraordinary considering how young she was. She developed this breakthrough at only 23 years old.
But the story takes a tragic turn. In 1916, Alice Ball died unexpectedly at the age of 24 before she had the opportunity to fully publish and secure recognition for her work. After her death, the president of the University of Hawaii, Arthur L. Dean, continued using her research.
But instead of crediting Ball, he published the treatment under the name Dean Method. For years, the medical community referred to the treatment using his name rather than hers,
which meant the chemist who solved the problem was almost erased from the history of the discovery.
Fortunately, the record was eventually corrected. Historians and scientists later revisited the research and confirmed that Alice Ball had developed the method.
Today, the treatment is widely recognized as the Ball method, and her contribution is honored as one of the earliest and most significant breakthroughs in the treatment of leprosy.
The University of Hawaii now commemorates her work with Alice Ball Day, and a plaque on campus recognizes her achievement. And beyond the historical recognition, the impact of her work was profound. Her method remained the primary treatment for leprosy for decades until the development of modern antibiotics.
Our ninth story shifts from early 20th century chemistry to the modern pharmaceutical revolution, and it centers on a scientist whose work helped change how medicines are designed. Her name was Gertrude Elion.
When Elion began her career in the 1940s, drug development was largely based on trial and error. Scientists would test thousands of compounds in the hope that one might show therapeutic effects. It was slow, expensive, and unpredictable.
Elion and her research partner, George Hitchings, believed there was a better way. Instead of randomly testing chemicals, they proposed designing drugs based on the disease's biochemistry.
Which sounds obvious now, but at the time it was a radical idea. Design the medicine around the biology of the disease instead of hoping something works.
Elion and Hitchings began studying how cells synthesize nucleic acids, the building blocks of DNA and RNA. Their insight was that if they could create molecules that interfere with these biochemical pathways, they might be able to stop the growth of harmful cells or pathogens.
Using this strategy, they developed a series of groundbreaking medications. Among them were treatments for leukemia, malaria, herpes infections, and drugs that made organ transplantation possible by preventing immune rejection. One of their most important discoveries was azathioprine, an immunosuppressive drug that revolutionized transplant medicine.
Another was acyclovir, which became one of the first effective antiviral treatments.
Which means that millions of patients around the world have benefited from drugs developed using Elion's approach.
Over the course of her career, Gertrude Elion was associated with more than 45 patents, reflecting the practical impact of her research. In 1988, she received the Nobel Prize in Physiology or Medicine, shared with George Hitchings and Sir James Black.
Unlike several earlier stories in our episode, this was a case where the scientific community eventually recognized her contribution at the highest level. And what makes her story even more remarkable is that Elion never completed a traditional doctoral degree. She entered research through laboratory work and built her career through innovation rather than academic titles.
Her achievements demonstrate that creativity and scientific insight can emerge through many different paths.
Our 10th story takes us into the world of material science and into one of the most remarkable inventions of the 20th century. A material that is incredibly strong, lightweight, and capable of saving lives. The scientist behind it was Stephanie Kwolek.
Kwolek was a chemist working at DuPont in the 1960s. At the time, the company was searching for new synthetic fibers that could replace steel in certain industrial applications, particularly for reinforcing tires. Researchers were experimenting with polymers, long chains of molecules that can be engineered to create strong and flexible materials. One day, Kwolek produced a polymer solution that looked unusual. Normally, polymer solutions used for spinning fibers were clear and syrupy. This one looked cloudy and thin.
Which in many laboratories would have been the moment someone said, "That experiment failed," and poured the solution down the drain.
But Kwolek had a strong intuition that the unusual appearance might mean something important. She convinced a technician to run the solution through the fiber spinning equipment anyway. The result was extraordinary. The fibers that emerged from the process were incredibly strong. Further testing revealed that the material had a tensile strength 5 times greater than steel, while remaining extremely lightweight. This new material became known as Kevlar.
Which today is used everywhere. Bullet-resistant vests, helmets, aerospace materials, sports equipment, protective gear for firefighters and law enforcement.
Kevlar's unique structure comes from the way its polymer chains align in parallel, creating strong molecular bonds that resist stretching and breaking. DuPont patented the technology and began commercial production in the early 1970s. Over time, Kevlar became one of the most important advanced materials ever developed. Perhaps most famously, it has been used to produce ballistic protective vests, which have saved thousands of lives around the world.
So this is one of those rare cases where a laboratory experiment turned into a global technology with immediate real-world impact.
Stephanie Kwolek eventually became one of the most respected chemists in industrial research. She was granted multiple patents and received numerous scientific honors, including the National Medal of Technology in the United States. Her work demonstrated how curiosity and persistence can lead to transformative discoveries. From an intellectual property perspective, Kevlar also illustrates the relationship between corporate research and patent systems. Large-scale industrial innovation often requires significant investment in research laboratories, equipment, and long-term experimentation.
Patents help companies protect those discoveries while bringing new materials to market. In this case, one chemist's curiosity about a strange-looking polymer solution led to the invention of a material that continues to protect people across the world.
Our 11th story also comes from the world of industrial chemistry and shows how sometimes innovation begins with an accident. The scientist behind this discovery was Patsy Sherman, an American chemist working at 3M in the 1950s. At the time, Sherman and her colleague Samuel Smith were researching new materials based on fluorochemicals, compounds that contain fluorine atoms and often have unusual chemical properties.
One day, something unexpected happened in the laboratory. A laboratory assistant accidentally spilled a fluorochemical solution onto a rubber shoe. Normally, a spill like that would simply ruin the shoe. But when they tried to wash it off, they noticed something strange.
The liquid had soaked into the surface, but it also seemed to repel water and stains. No matter how hard they tried, the stain would not behave like a normal spill.
Which must have been one of those moments where scientists pause and say, "Wait, that is not supposed to happen."
Sherman and Smith began investigating the phenomenon more carefully. They discovered that fluorochemicals had the ability to repel both water and oil-based substances, something extremely rare in chemistry. Most materials repel one or the other. Fluorochemicals could resist both.
After extensive research, they developed a treatment that could be applied to fabrics and other materials to protect them from stains.
That product became Scotchgard.
Which anyone who has owned a couch, a carpet, or a jacket has probably encountered at some point.
Scotchgard quickly became one of the most successful consumer protection products in the world. It allowed fabrics to resist spills, dirt, and stains while maintaining their original appearance. Patsy Sherman was granted multiple patents related to the chemistry behind the invention and became one of the most prolific female inventors of her time. Over her career, she held 13 patents, many related to fluorochemical applications.
Which is impressive in any era, but especially in the mid-20th century, when women were still underrepresented in industrial research laboratories.
Sherman's work also demonstrates an important aspect of innovation. Many discoveries begin with unexpected observations. The difference between a failed experiment and a breakthrough often lies in whether a scientist chooses to investigate the anomaly.
Our 12th story begins on a snowy day in New York City in the early 20th century. The year was 1902. A woman named Mary Anderson, a real estate developer and entrepreneur from Alabama, was visiting the city when she noticed something frustrating about the streetcars.
The drivers had a serious visibility problem. Snow and rain would collect on the windshield, making it difficult to see the road. In order to clear the glass, drivers had to stop the vehicle and manually wipe the windshield. Sometimes they would even open the window and try to clear the snow by hand while driving.
Which sounds extremely unsafe and extremely inconvenient.
Mary Anderson looked at the situation and thought there had to be a better solution. When she returned home, she designed a mechanical device that allowed the driver to clear the windshield without leaving the vehicle. Her invention consisted of a rubber blade attached to a lever inside the car.
When the driver moved the lever, the blade would sweep across the windshield, removing rain, snow, and debris.
In 1903, Anderson received a United States patent for her invention. It was one of the earliest patents for what we now call the windshield wiper,
which today feels like one of the most obvious pieces of automotive equipment ever invented. You literally cannot drive in bad weather without it. But at the time, automobile manufacturers were skeptical.
Many companies believed the device would distract drivers or that people simply would not need it. As a result, Anderson struggled to license the technology. Her patent lasted for 17 years, which was the standard patent term at the time. But by the time the automobile industry began widely adopting windshield wipers, her patent had expired.
Meaning the entire automotive industry ended up using the concept without paying royalties to the inventor.
Today, windshield wipers are standard equipment on every vehicle in the world. The global automotive industry relies on a technology that began with Mary Anderson's simple but brilliant observation during a snowy streetcar ride. Her invention eventually became recognized as one of the most important early safety innovations in transportation.
Which shows that some inventions do not look revolutionary at first, but once they exist, the world cannot imagine functioning without them.
From an intellectual property perspective, Anderson's story highlights the importance of timing and commercialization. A patent can protect an invention, but if the market is not ready to adopt it during the patent term, the inventor may never see the economic benefits. Still, Mary Anderson's idea reshaped transportation safety. Every time a windshield wiper clears the rain from a driver's view, it is quietly repeating the motion she imagined more than a century ago.
Our 13th story takes us into the world of board games, economic theory, and one of the most famous intellectual property stories in popular culture.
The game is Monopoly. Millions of people around the world have spent evenings buying properties, collecting rent, and occasionally flipping the table when someone lands on boardwalk with a hotel. But long before Monopoly became a commercial success, a woman named Elizabeth Magie created a game with a very different purpose. In 1904, Magie patented a board game called the Landlord's Game.
Magie was a writer, activist, and supporter of economic reform ideas inspired by economist Henry George, who argued that monopolies over land and property created social inequality. Her goal was not simply entertainment. The Landlord's Game was designed as an educational tool. It demonstrated how wealth could accumulate through property ownership, while others struggled to keep up.
So the original idea behind the game was actually a critique of monopolies, which is ironic considering the name the game eventually became famous under.
Magie's patent described a board where players moved around collecting rent from properties. But the game included two different rule systems. One version showed how monopolies concentrated wealth. The other version illustrated a more cooperative economic system where prosperity could be shared.
In other words, the game was meant to teach economic principles. Over time, the game spread informally. People copied it, modified the rules, and shared homemade versions across universities and communities.
Then in the 1930s, a man named Charles Darrow encountered one of these versions. Darrow adapted the game and presented it to Parker Brothers, a major board game publisher. Parker Brothers eventually purchased the rights and began selling the game commercially under the name Monopoly, and quickly became one of the most successful board games in history.
For many years, the public narrative presented Charles Darrow as the sole inventor of the game, but historical research later revealed that the core concept originated with Elizabeth Magie decades earlier. Parker Brothers eventually purchased Magie's original patent rights for the Landlord's Game, but the commercial success and public recognition largely centered on the Monopoly brand and Darrow's story.
It raises questions about how inventions evolve as they move through communities, adaptations, and commercial markets.
Today, historians widely recognize Elizabeth Magie as the true originator of the concept that became Monopoly.
Our 14th story begins in Hollywood, which might not be the first place people expect to find a groundbreaking communications technology. The central figure is Hedy Lamarr, an Austrian-American actress who became one of the most famous film stars of the 1930s and 1940s. Lamarr was celebrated for her beauty and her roles in major motion pictures. But outside the film industry, she had another passion.
She loved engineering and invention. During World War II, Lamarr became concerned about the vulnerability of Allied naval torpedoes. At the time, many torpedoes were guided by radio signals.
The problem was that enemy forces could intercept or jam those signals, making the weapons ineffective. Lamarr began thinking about how to prevent that interference. And this is where the story becomes truly unexpected. Her collaborator was not another engineer. It was a composer.
Lamarr worked with George Antheil, an avant-garde composer known for experimenting with synchronized musical instruments. Together, they came up with a remarkable idea. What if the radio signal controlling the torpedo constantly changed frequencies?
If the transmitter and receiver switched frequencies in perfect synchronization, the signal would be extremely difficult for an enemy to detect or jam. They designed a system in which the signal would rapidly hop between different frequencies following a synchronized pattern.
The concept became known as frequency hopping spread spectrum. In 1942, Lamarr and Antheil received a United States patent for the technology,
which today sounds like a cornerstone of modern wireless communication. But at the time, the U.S. Navy did not adopt it.
The Navy considered the technology too complex to implement during the war. As a result, the patent was never commercialized during Lamarr's lifetime. The patent eventually expired. Decades later, however, engineers began developing wireless communication systems that used similar principles. Frequency hopping and related spread spectrum techniques became fundamental to technologies such as Wi-Fi, Bluetooth, and GPS.
In the 1990s, historians and engineers began recognizing the significance of her patent. In 1997, Lamarr and Antheil received the Electronic Frontier Foundation Pioneer Award, acknowledging the influence of their invention on modern communications technology. The most fascinating examples of an idea that was decades ahead of its time.
Our 15th story moves from laboratories and inventions into the world of art, copyright, and one of the most unusual courtroom moments in intellectual property history. It centers on Margaret Keane, an American artist whose paintings became famous for their distinctive style. Her works featured children and figures with large, expressive eyes, a visual signature that quickly became recognizable.
In the late 1950s and early 1960s, these paintings became extremely popular. They were reproduced on posters, prints, and postcards and sold widely across the United States. But the public believed that the artist behind the paintings was Walter Keane, Margaret's husband at the time.
According to Margaret, Walter had convinced her to allow him to claim authorship of the paintings. At the time, he managed the business side of the operation and argued that a male artist would be taken more seriously in the art market. So for years, the paintings were sold under his name. As the works became commercially successful, the public narrative around the Keane paintings centered entirely on Walter as the artist.
Margaret continued producing the paintings, often working long hours in the studio while Walter handled exhibitions, marketing, and publicity. Eventually, Margaret left the marriage and began publicly stating that she had been the real artist all along. Walter denied her claims,
which meant the dispute eventually ended up in court.
And this is where the story becomes unforgettable.
In 1986, the case went before a federal court in the United States. The central question was simple but profound. Who actually created the paintings? To resolve the dispute, the judge proposed an unusual solution. He asked both Margaret and Walter Keane to paint in the courtroom. They were given materials and time to demonstrate their artistic ability. Margaret began painting immediately. Walter declined, claiming that he had a shoulder injury that prevented him from holding a brush.
Which is not the kind of explanation that tends to strengthen your position in a copyright case about painting. Margaret completed a painting in the courtroom in less than an hour. The demonstration, combined with other evidence, helped persuade the court that she was the true creator of the artworks. The jury ruled in her favor and awarded damages for defamation. More importantly, the case publicly confirmed that Margaret Keane was the real artist behind the famous Big Eyes paintings.
Which makes this case one of the clearest examples of authorship being tested in a courtroom.
Literally with a paintbrush.
Our 16th story takes us to the very beginning of the film industry. Before Hollywood, before blockbuster studios, before the idea of cinema as a global entertainment business. At the center of this story is Alice Guy Blaché, one of the earliest filmmakers in history and arguably the first woman film director.
In the late 19th century, moving pictures were still a technological curiosity. Most early films were short recordings of everyday scenes, trains arriving at stations, people walking in the street, simple moments of life. Alice Guy saw something different. She believed film could tell stories.
While working as a secretary at the Gaumont Film Company in France in the 1890s, she persuaded her employer to allow her to experiment with filmmaking.
In 1896, she directed The Cabbage Fairy, one of the earliest narrative films ever made. Instead of simply recording reality, the film staged a fictional scene with characters and storytelling. Over the following years, Alice Guy directed or supervised hundreds of films.
She experimented with narrative techniques, synchronized sound experiments, hand-colored film, and complex storytelling structures. Later, she moved to the United States and founded her own production company, Solax Studios, becoming one of the first female studio owners in the film industry. At its peak, Solax was one of the largest film production companies in America.
But as the film industry evolved and large studios began dominating production, many early works were lost or misattributed.
For decades, numerous films directed by Alice Guy were credited to male colleagues or simply listed without clear authorship. Some historical records even omitted her name entirely. As film history was written in the 20th century, the narrative often focused on male directors who worked in the same era.
Alice Guy's contributions faded from mainstream recognition.
In recent decades, historians and archivists have begun restoring her legacy. Researchers have identified and catalogued many of the films she directed and helped establish her role as one of the most important pioneers in cinematic history.
Today, Alice Guy Blaché is recognized as a filmmaker who shaped the foundations of narrative cinema long before Hollywood became the center of the industry. When you listen to all these stories together, a pattern starts to appear. Different centuries, different countries, completely different fields, and yet the same thing keeps happening. A scientist notices something unusual in a laboratory. An inventor tries to solve a practical problem. An artist develops a new way of seeing the world. Curiosity starts the process, and persistence carries it forward. What is remarkable is how many of the discoveries we discuss today quietly shaped everyday life. From medicines that save millions of lives, to materials used in modern technology, to creative works that transformed culture. These breakthroughs are now part of the fabric of the modern world.
And that is why recognition matters so much. Innovation is not only about the idea itself, it is also about the connection between the idea and the person who made it possible. Intellectual property systems, whether patents or copyrights, help preserve that connection. They allow discoveries and creations to remain linked to the people behind them.
Which means intellectual property is not just a legal framework. It is also a way of remembering how progress happens. It tells the story of who experimented, who invented, and who imagined something that had never existed before.
And when we revisit stories like the ones we explored today, we realize something important. Women have always been present in the history of innovation. They were in the laboratories, the research institutes, the workshops, and the studios. Sometimes they were recognized immediately. Sometimes the recognition came much later.
But the ideas endured. the discoveries endured, and eventually the stories began to surface.
International Women's Day is a moment to reflect on that reality. Progress in science, technology, medicine, and the arts depends on the diversity of minds that participate in it. When more people are able to ask questions, test ideas, and explore new possibilities, the horizon of innovation expands.
And the stories we share today show exactly that. Innovation often begins with a simple observation, a pattern in maize kernels, a signal in radio data, a problem with rain on a windshield, a new material forming unexpectedly in a laboratory.
Small moments of curiosity that eventually lead to discoveries capable of changing entire industries and shaping how society functions.
So today we celebrate the scientists, inventors, and creators whose work helped build the world we know.
And we remember that every new discovery begins with someone willing to ask a question that no one else has asked before. Happy International Women's Day.
Thank you for listening to Intangiblia, the podcast of intangible law, plain talk about intellectual property. Did you like what we talked today? Please share with your network. Do you want to learn more about intellectual property? Subscribe now on your favorite podcast player. Follow us on Instagram, Facebook, LinkedIn, and Twitter. Visit our website www.intangiblia.com. Copyright Leticia Caminero 2020. All rights reserved. This podcast is provided for information purposes only.